Hello everyone. So today I'll be discussing about the biology diagrams of class 10 term 2. Okay. So before that, let me introduce myself. I am Vani Kari, biology educator at Unacademy. I am an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So let me just check if I'm audible. Okay. So join with us in Telegram. So for homework and class notes, you can join the channel Vani Kari ICSE Class 10 Biology. So you can search it in the Telegram and you will be finding this. You can join this one. And Telegram channel of Unacademy, ICSE and ISC both. So this you will link you will find in the description box. Okay. So you can click on the link and join the channel for updates regarding the classes again. Hi Siddhi, good evening. Okay. So Unacademy free class or special class also we call them. So the features are basically again like here. So you can have a live real-time interaction using the chat and the emojis. Ask questions using the question tab. Okay, so there is a separate question tab. So if lot of students are there, so there is a chance that your question might get lost. So you can type in your question, attach an image in the question tab and send it to the educator during a live session. So they'll be getting a notification and will be clearing your doubts. Okay. Hi Crazy Tech, good evening. Okay, then there is live poll option also available. So what you can do is there is a option that you will be getting. So the question and the options will be seen on the screen. So you can choose the option A, B, C, D, and after that we will be sharing the leaderboard as well, so that you can know your rank and you can compete with your friends as well. So now Unacademy subscription features. So you can learn live from the comfort of your home, unlimited access to all the courses. So there are different courses that are going on. So different educators have come up with different courses. So you will have access to all these courses once you go for the subscription. Then top educators of India are going to take the classes in one platform. Regular doubt clearing session. So after about every 3 sessions, the 4th session that we conduct will be a doubt clearing session. So any doubts you have related to the topics that are previously covered, so you can clear your doubts in this session. Then complete coverage of a syllabus is done, mentorship and guidance is given, study material is given, practice test is given, live test series, batch courses. So you will have access to the batches. So there is a batch in Hindi, there is a batch in English, there is a batch in English. So you will have access to all these batches. Then there is daily practice section, weekly mock test series, live quizzes, daily MCQ and subjective test series will also be there. So now with respect to now for class 10, okay, so you can try with this one. So there is a 3-month offer which is there. So if you want to try how exactly this works, so you can go for 3 months of subscription. And if you are planning to complete class 10 with Unacademy, so you can go with 12 months of subscription as well. So now there is an offer. So with this referral code Vani YT, so you will be getting 20% discount. So use the code Vani YT. So with this code you will be getting 20% discount instead of 10% discount. Okay, so this offer is valid today and tomorrow. This offer is there, so hurry up and take the subscription so that you can have access to all the batches. So in a regular manner the classes will be done, so complete syllabus can be covered there. And yes, so let's have a look at the circulatory system diagrams. So you know the diagrams are given. Okay, so the diagrams are basically the heart, and they can ask you to draw a diagram of artery and a vein as well. Okay, so you should be able to draw this one, so you can see the differences there. Okay, so external layer, the connective tissue is given, then you can see, so this one is the external layer. So it is like, you know, a thicker one is present over here. So here also it is not so thin, but it is somewhat thicker. The muscular layer that you need to observe, so let's have a I'll draw it and explain, so you can have a look at the muscular layer also. So here it is much more thicker as compared to the vein. Then elastic fibrous, so this is basically with respect to smooth muscle only. Then there is endothelium, so endothelium will be the same anyway. Okay, so that is only one single line thickening will be there. So let's see the structure, and the other structure which is mentioned here is heart. Okay, so these two structures that you need to draw, and they can ask you, you know, chordate tendinase being labeled here, then you know papillary muscles also that you need to represent. So let's have a look at this one now. So let's see the chamber. Okay, so first we will be drawing the right auricle. Okay, so this is now a right auricle, and then right ventricle. So first you are drawing these chambers here. Okay, so same way towards the other side. So left auricle you are drawing, and then there is left ventricle you will show. Okay, so this one is the left auricle, and then there is left ventricle. One second. Okay, so left ventricle you draw, and after that we will need to draw the muscular covering which is there. Okay, so muscular covering, you know that a double layer of protection is there. So that is, you know, the pericardial membrane. All right, please explain which is tunica externa, interna and media. My teacher taught me to write it like this. Okay, so this one is okay. So now we are asking about the tunica externa. So this outermost layer which is there. So this is tunica externa. All right. So this one is the tunica externa. So this can be the middle one. So this is the tunica media, and this one what you see endothelium. So this is a tunica interna. Okay, so this is the one in case of artery and vein. So instead of drawing, writing external layers, so you can also write it like this. But if since your teacher has asked you, but in your ICSE anyway you can use this for practice, but in ICSE evaluation, so when they ask you to draw, so you can label this, but make sure that you are writing these labels also. You can write this also, no problem, but make sure that you are drawing, naming or labeling these also. Okay, so this is very important, so make sure that you are going with this one. So just a second, so there is some problem with my video today. One second, let me just check. Okay, all right, so let's get started with the heart structure that I was showing. I hope it is clear, Siddhi. So now you need to draw the two-layer protection for this one. So that is a pericardial membrane. Okay, so there is a muscular covering that you will be drawing. So this is now the muscular covering that you need to show. Okay, so one second, this become flat. Okay, so now this is a muscular covering which is there. Okay, so it is not just one covering, so you need to draw two coverings that for this one. Okay, so peritoneum, when the pericardium, when we mention, so it is the two covering that we need to draw. So because the external one is there and the internal one, and in between you will be showing the pericardial fluid, so we need to draw two coverings for this one. Okay, so this is now a heart with the two covering, the external one and the internal one, and in between that you can find a pericardial fluid. So there will be a pericardial fluid. So let me just show you this. Okay, what happened here? All right, so now this is the structure. Okay, so in between this you will be showing a pericardial fluid which is present. Okay, so this is the pericardial membrane, so this is the pericardial fluid. So instead of drawing it everywhere, so instead of filling it everywhere, so I'll just fill it in a part so that you can understand. Okay, So now this is the structure. So I'll just label each one of them. So this is a pericardium. Okay, so pericardium is basically a pericardial membrane. So both of these, one is outer one and the other one is inner. So pericardium, all right, or pericardial membrane, or pericardium also can be mentioned. And this one inside the fluid, what you see, so this is a pericardial fluid. Okay, so this is a pericardial fluid. So let's see the labeling for the right auricle, the right ventricle. Your endocrine video is not available in playlist. What is the function of pericardial fluid? So function of pericardial fluid is it is basically lubricating the membrane, so it will avoid the friction between these two membranes. So there is the outer membrane and the inner membrane, so it will it will avoid the friction. That means it is lubricating. Another function is it will act as a shock absorber, and also it helps the keeping these two membranes together. Okay, so that is the function with respect to mechanical injury. So protection from a mechanical injury also we can mention, or a shock absorber can also be mentioned. So this is the function of pericardial fluid. So endocrine video is not available in the playlist. I think it will be there, so you can just search it once. So it is endocrine system, some hormone you are asking, the one which I had taken recently, or the diagram based question you are asking. I'm not getting which video exactly. So you can just tell that. I'll check it out. Okay, so I'll check it out. So if it is there, I'll anyway send. I think no videos are removed, so every video will be there. So I'll be sending the link on my Telegram channel itself. So you can join Vani Kari ICSE class 9 and 10 biology there. Okay, so now this is the structure. So this wall, what you find, so this is actually the muscular covering, and you know that here I have drawn a thin muscular. So let me just show you this one. Okay, so this side part, what you see, I hope this is visible. So this is thicker, so this is towards the left ventricle, and this one is the left auricle. So this is right auricle and this is right ventricle. Okay, so towards this side I have made it thicker, so you know that the ventricle wall will be thicker. Okay, so that you need to make sure that while you are drawing, also make sure that you are drawing left ventricle side, the thicker muscular wall you are drawing, because the pumping action will be done by the left auricle, so that the blood, oxygenated blood, will be entering the aorta part. Okay, so from here you need to draw the the pulmonary artery. So here you are showing the pulmonary artery now, which is actually carrying the deoxygenated blood. Okay, so before that, let me just show you the tricuspid valve and bicuspid valve, and then I'll go with that. Okay, just a second. Okay, so now here you can find a tricuspid wall. Okay, so there is a tricuspid wall which is held on. So let's say here the papillary muscles are here. Okay, so this is now a papillary muscle which is labeled here, so you can check this one. So papillary muscle is there, and the chordae tendineae is holding on to this one with the walls. Okay, so you can draw this one. So what you need to do is you can just hold these structures with this one over here. Okay, so this is basically, it is holding on to the papillary muscle. So on this side you will be showing the bicuspid valve. Okay, so this one is a bicuspid valve, and here also you will be showing the papillary muscles again. So they are holding on to the structure. Okay, so this is a chordae tendineae. So now let's show this one. So this one is now a tricuspid valve. Okay, so there is tricuspid valve, this thread like structure what you see. So this is a chordae tendineae. Okay, so this is a chordae tendineae, and so you can see this one, the chordae tendineae. Okay, so chordae tendineae is there, and then this muscle what you see, so this is a papillary muscle. Okay, so this is a papillary muscle. So same towards the other side also you can see that, and from here you will be showing the pulmonary artery. Okay, so pulmonary artery arises from here. Just a second. So pulmonary artery arises from here, and this is gonna branch. All right, so this will be branching on the upper side. So basically it is dividing towards the right side and the left side. So this one is now a pulmonary artery which is entering from the right right ventricle, and there is semilunar valve at the end of this pulmonary artery. So we will just label this one now. So this part what you see, so this is a semilunar valve. Okay, so this is a semilunar valve, and here you can see a pulmonary artery. Okay, so this one is pulmonary artery. Okay, and towards the other side, so that is basically with respect to, okay, so the other side, so you can find a aorta. So arising from here you can find a aorta. So let's just draw this one. So here is this aorta which will be joining here, which is actually forming a structure as such over here, and then it is continuing down here. Okay, so here you can find 3 openings which are there, so it will be joining the upper parts of a body. Okay, so that means this is connecting, this is supplying a oxygenated blood to the, so from here you can we can just show it as such. Okay, so this is now a aorta. Okay, so at the base of the aorta, so let me just make it here, so you can find again a semilunar valve there. So semilunar valve is present here also, so we will just connect this one. Okay, so aortic pulmonary valve, pulmonary semilunar valve. So this one, this part is actually the aorta. Okay, so which carries the oxygenated blood. Okay, yes, Siddhi, yes, what is the doubt? Process of clotting, yes, I'll explain it. Okay, I'll explain it after some time. Okay, so I'll just complete the structure of artery and vein. I'll just explain it. Okay. Okay, so now aorta is done. So now what else do you need to mention here? So you need to show the pulmonary vein. Okay, so pulmonary vein which is joining here. So you still need to show the inferior vena cava and the superior vena cava. So both the side, make sure that you are showing a pulmonary vein. Even though this pulmonary vein here we are showing as such. Okay, so it is not entering the right auricle. So this will be joining towards this side. So you can show the pulmonary vein which is actually joining in here. So this is basically, it is entering to the left ventricle, left auricle. Okay, so this is coming towards the left auricle. So from this side also, so from this particular side also, 2 of them you will be showing, but they are not entering the right auricle. So this is like basically they are joining in here itself. So I am just showing a dotted line. So they will be joining the left auricle itself. Okay, so this is the pulmonary vein. So this one I can label this side also. So I'll just label it here. Okay, so this is now a pulmonary vein which is bringing a oxygenated blood to the left auricle. So it is coming from this side, that is from the right lung and from the left lung, but it is not emptying its content into the right auricle. So this is emptying its content into the left auricle itself. Okay, so now other than that, so there is a superior vena cava and inferior vena cava that we need to show. So now here, this one over here. Okay, so I'll just show it here itself. So here you can see, okay, one second. Okay, so this part what you see, so this is exactly the inferior vena cava. Okay, and here you can see, so I'll just erase this part. So here you can see the superior vena cava. Okay, so this part is the superior vena cava through which the blood is entering, that is from the upper body parts. Okay, so from upper body parts the blood is coming. So you can just erase this part. Okay, so whenever such thing happen, so you can erase this part so that it will be understood later on. Okay, and now from lower body parts, so there is actually the deoxygenated blood that is coming. So this is the inferior vena cava. Okay, so this is IFC. Okay, one second. Inferior vena cava, and this one is a superior vena cava. It can also be called as anterior vena cava. Okay, so this can also be called as a anterior vena cava, and this can also be called as a posterior vena cava. So this is the structure that we have done. So somebody has asked me, what is it? Process of clotting. Yes, that is done. I'll explain about it. And what is this? Chordate tendineae. Okay, I'll explain. So basically, chordate tendineae will regulate the opening and closing of these bicuspid and tricuspid wall. So here you can see this one is bicuspid valve, okay, so which is held on to this papillary muscle. So here you can see this papillary muscle, a chordate tendineae. So bicuspid valve and tricuspid valve regulation, so that is basically opening and closing. So with respect to the chordate tendineae, so as there is a muscle contraction that can occur, so you know this chordate tendineae will be regulating the opening and closing with respect to the bicuspid and tricuspid valve. Okay. Yeah, so this is the structure of a heart that you can draw. Okay, I think all the other parts. So septum you can show it here. So I'll just explain that. So this part what you find, so this part in the center, okay, which is actually not labeled. So this is basically the separation that is occurring between the ventricles. So that can be labeled as a septum. So this is a interventricular septum. Okay. So next one is artery and a vein. Okay, so now for artery and a vein, so nothing is there, so this is very easy. So first you will be drawing a circle. Okay, so thicker wall that you need to draw here. So now with respect to, you can see this one over here. Okay, so thicker one you will draw, that is the connective tissue is thicker, but in this case also, so you will be drawing the vein which is slightly somewhat like this, you can draw. Okay, and the connective layer which is there, so that is not so thicker, so this is somewhat a thin itself. So we will draw one more outer covering for this one. So as compared to the other one, here we can see this is slightly thinner. Okay, so here thicker external layer or tunica externa is somewhat thicker as compared to the vein. The muscular layer as well, so you can see now this one over here. So I'll be just showing this one. So muscular layer which is there. Okay, 1 second, I'll erase this part. Okay, so this muscular layer what you draw, so that also should be thicker. So this is completely, you are showing a muscular layer as such. So this will be a completely a muscular layer itself, and here you can just show this dotted as such. Okay, so in this case you can draw the muscular layer. So here it is thin again. Okay, so this muscular layer is very thin as compared to this artery. So artery, a thicker layer is there. So this line what we are showing here, so this is basically the muscular layer. So this one over here, so it is somewhat thinner as compared to the artery. Okay, so this one you will show the dotted appearance, so you can show this dotted, and the other one, so you can see this layer now, right, the endothelium. You need not draw anything here. So this layer what you have drawn, so here itself you can just show it like this. Okay, so just draw it like this. So that means endothelium is basically one single layer. Okay, so let's say this is a epithelial tissue, so it is not having many thicker layers. Okay, so that means piles of layers are not there. Endothelium, when we mention, just one small, one squamous epithelial will be there. So if I am taking this layer and flattening this, so only like this you can draw. Okay, so this is the layer, so only one layer of a cell will be there. So for this also, so you can draw just one layer of a cell. So this is one layer of a cell that you will be showing. Okay, so this is one layer of a cell. So now if we have to make it this one, so now this is the tunica externa or the external layer. Okay, so this one dotted one is the external layer. So this one, the line like what you see, so this is exactly the thicker one in case of artery, and here it is a thinner one. So this one can be your middle layer. So there is a middle layer which is basically made up of a smooth muscle. Then you can see this one, so this structure, so only one line you need to draw, and that is the endothelium. So instead of a epithelium, what is epithelium? So it is a epithelial lining which we have, right? So for our the cell also, skin cell is epithelial, so it is lining on the outside. Now you know inside the blood vessel it is lining. So now this is a blood vessel. Inside the epithelial cell is lining as such. So instead of a epithelium, now we call it as a endothelium. It is the epithelial cell itself which is lining on the inner side, and it is just one single layer of a cell, so that is why it is a endothelium. So you can just draw it endothelium as such. And now you can easily understand that the lumen over here is wider and the lumen here is smaller. Okay, so always make sure that you are drawing a smaller lumen, or narrow lumen in case of a artery, and a wider lumen in case of a vein. So here the lumen is wide, and here in this case the lumen is narrow. So narrow lumen you need to draw, and here it will be the wide one. Okay, yes, artery and vein structure they will ask you to draw, or if not they can also ask you to they can just give you a diagram based question. So from diagram based question you need to recognize which is the artery and a vein, so that will be done based on the structure which is mentioned. Okay, will you bring one short revision on 10 ICSE? One short revision on class 10 ICSE. I think you only had asked previously. Yes, I'll do that. Okay, I'll do that, but not tomorrow. Tomorrow I have other classes. Maybe we can do it on this thing. I mean, tomorrow is which day? 16th, right? So 17th I'll do it. Okay, so 17th I'll do that. I'll assign it now only. Okay. Okay. So moving on, yeah, so Siddhi was asking me about clotting process, right? So I'll just explain that. The tissue is damaged. So let's say this is the skin, okay? So skin is actually damaged, okay? So skin damage is there. So whenever a damaged skin is there, so the blood which is inside here, so here the blood vessel was there, okay? So this blood vessel is also damaged. So in that case what can happen? So the tissue which is surrounding here, the damaged part, so the tissue is affected. So here it will be made up of certain cells, right? So all these cells, and here the damaged cells are there. So these damaged cells, okay, so damaged cells, and along with that the platelet which is here, okay, so here the platelet will be there in the blood vessel. So that platelet, so together they'll start releasing a clotting factor. So thrombokinase is released. Okay, so once the thrombokinase is released, so this thrombokinase will act on the prothrombin. Okay, so this will be, prothrombin is actually converted into thrombin here. Okay, so this thrombin is the active one. So this is all present in the plasma itself. So this is an inactive one. This thrombokinase will act in presence of a calcium ion. Okay, so calcium is required, then only this prothrombin will be converted into thrombin. Then this thrombin will act on what? It will be acting on the fibrinogen. So fibrinogen is actually soluble. Okay, so soluble it will be. Now this fibrinogen will be made into a fibrin which is actually insoluble one. Okay, so insoluble fibrin, once it is formed, so let's say this insoluble fibrin is a thread formation. So like this thread will be formed. Okay, once it is acted by the thrombin. So this will also require a calcium here. So calcium is required here as well. So once these threads are formed, so these threads will form a mesh here at this point. Okay, so that means a blood plug formation will occur. So because of this blood plug or a clot, escape of blood cells will not occur outside. So or the plasma also is not escaping or going out, or the germs which can take entry. So germs also cannot take entry. So germs cannot enter, and the RBCs, WBCs, so they are not going out as the blood clot formation is occurring. Okay, so I hope it is clear. So let's get with the excretory system. I'll just explain that. What will be the time for one short session? So one short session, no, it will be I'll try to keep it 17th. I need to check my timetable. Okay, I'll check the timetable and let you know. only on nervous system and endocrine system. Okay, only on nervous system and endocrine system. You want me to do the revision? Okay, I can do that. Let's see. Okay, I'll anyway inform you all. I can't inform you timing now itself. I need to check that, and I'll let you know about that. Okay. So now this is the structure that you have here. Okay. So that is the aorta structure. One second. Okay. So for aorta structure, I mean aorta is there, kidney is there, renal artery. So how you can draw this one? Okay. So let's have a look at this. So there is a kidney. So this one is a right kidney. Okay. So left kidney also that you need to draw. So left kidney you will be drawing slightly on the upper side. Okay. So upper side you need to draw. So you can, lightly, slightly you can show as such also a very little, or now this can be too much of a, difference might be there. Better to show it this way also, so that they can understand that that the left kidney is on slightly higher as compared to the left one. Okay, I mean right one. So now this is a right kidney. Okay, so this is a right kidney and this one is a left kidney. So on top you can show a adrenal gland. Okay, so that can be shown. So you can just make a cap like structure onto this one. Okay, so here also make sure that you are drawing a cap like structure onto this one. Okay, so this is done. So once that is done, so what you can see here, so there is a hilum, the ureter which is attached at the hilum. So let's draw this one now. Here you can see a ureter. So this one over here is now a ureter is here. Okay, so this one is ureter, which is actually projecting into, so this is projecting into the urinary bladder. So on this side also you can show this ureter. Okay, so there is a ureter which is again projecting into the urinary bladder. So let's see here, I'll just show this urinary bladder over here, just a second. Okay, so now this structure what you see, so this is a urinary bladder. Okay, so urinary bladder you will be showing a opening over here. So this is the opening which is there, which is again guarded by a sphincter muscle. So you can just show the sphincter muscles as such. Okay, so let's label this one. So this is a right kidney, this is left kidney, and this part is adrenal gland. Okay, so this is adrenal gland. Then you can see a ureter over here. So this diagram is not yet complete. Okay, so you still need to draw the aorta and vein also. So this one a ureter, so this one is the left ureter, this one is a right ureter. Okay, so this is right ureter, this is left ureter, and this one is urinary bladder. Okay, so urinary bladder is present. So after that you can see this part or this opening which is there. So this is a urethra. Okay, and this one what you see, so this is a sphincter muscle which on the impulse from the brain, so this is gonna open up. So this is a sphincter muscle. So the relaxation and contraction of sphincter muscle will occur. So based on that only the urinary bladder will be opening or leaving, micturition is gonna occur. So that is urination can occur based on the contraction that is or relaxation that is occurring in the sphincter muscle, which will mainly occur with respect to the impulse from the brain. And this one you can see projecting ureter is there. So you always need to draw the projecting ureter. So they are not straight here as such. So they are not completely straight. So there is a backflow which can occur. Okay, so now here as they are projecting, so backflow will usually avoided. So now let's have a look at this one. So you can see the aorta. Okay, so aorta is divided into renal artery. So let's show this one now. So now here you can see a aorta. Okay, so this aorta is divided and it is forming a renal artery. So you can see this one. So here is the formation of renal artery, and then it will continue down here. Okay, so this part I need to delete in just a second. Okay, so this one is now a aorta. Okay, so I'll just label this one. So this is a aorta. And now from here, so this is the direction, a movement of blood which is entering here. Okay, so blood can enter here also. So this is basically connecting other parts. Okay, so this is basically connecting the other parts of the body. Okay, so now there is blood which has to leave from here. So the blood which is leaving, so this will be leaving through the vein. Okay, so there is a vein, you can see here also. So there is a aorta and a vein. So we can show the vein here itself. So vein is there. So from here the vein is joining, and we can show one more over here, just a second. Okay, so from here it will be joining here, that is at the inferior vena cava it will be going. Okay, so from here this is the direction of the blood with respect to vein. So now it will be joining the inferior vena cava. Okay, so this is the inferior vena cava, it is going to join later on. But you know, renal vein is forming from both of these, and it will be joining the inferior vena cava. So let me just label these two parts now. So this one, what you see, so this is the renal vein. Okay, so this is the renal vein, and the other one what you see, so that is the renal artery. So I'll just label it here itself. So here you can see a renal artery. So there is a renal artery, renal vein, aorta and a IVC. So this is basically inferior vena cava. So this structure also again easy. Okay, all you need to do is draw the structure of a kidney. So you are drawing a kidney here. So this one is now a kidney. One second, it became too curved. So much of curve anyway is not required. Okay. So now you know there is the kidney which is there. Okay, so two layers of covering. So two coverings you need to draw for a kidney as well, as you can see here. So there is a two covering. So don't just draw one covering for this one. So for this also you need to draw two coverings. Okay, so this will indicate the presence of cortex and medulla can also be indicated with this one. So two coverings you will be showing. Okay, so here with that covering, so you can see that the cortex region is separated and the medulla region is separated. Why opening of urethra? So opening of urethra, one second. So it is because of the sphincter muscles. Okay, so sphincter muscle is the one which is regulating the blood flow, I mean urine flow, sorry, not the blood flow, urine flow. Okay, blood also can flow only if there is, you know, infection is there, so associated with the urine there can be blood flow also, but usually through the urethra it is the only urine that will be passing out. Okay, so now under the relaxation of the sphincter muscle, so once the urine is 2/3 full here, so let's say urine is 2/3 full in this case, so you know the impulse is received by the brain for the relaxation of sphincter muscle. So as soon as the sphincter muscle relaxes, so urination or micturition can occur through the urethra. Okay, yeah, so I was just drawing this one. So now this is a kidney that you are drawing, and you need to make a partition here. So there is the outermost one is a cortex layer. So this one over here, so this will show you the cortex one. All you need to do is a dotted structure. Okay, dot one that you need to draw. Okay, so why there is dot? Anybody has any idea on this one, why this appears as dotted structures? I mean structure when we observe under a microscope or something, or maybe directly also. Any idea why this is dotted? So now this is the dotted region. So this one is actually the cortex. Okay, so renal cortex, and this one is actually capsule. So even though it is not mentioned in your textbook, okay, so outer covering which is there, so this can also be called as a renal capsule. So one more covering is there for your kidney. Then you know this part what you see, so this is a medulla part. Okay, so medulla part what you can show, so you can just draw the pyramids as such. So So what you can do is just draw the pyramids as such here. Okay, so once you draw the pyramids as such, so make sure that, so you can just draw the pyramids over here. Okay, so once you draw the pyramid, make sure that you are adding a lines into it. So just show the line appearance. So this line is basically obtained, so these line appearance you can see only because the connecting loop, collecting duct and the Henle's loop, so they are giving this striated appearance. Okay, so now this is the pyramid that you see. So this one is a pyramid, and this apex what you see, so I'll just show you this. So this apex of each of these renal pyramid which is there, so this apex is called as a papilla. Okay, so now I'll just label this part. So this is now a renal pyramid. Okay, so this is a renal pyramid, and apex of renal pyramid is the papilla. Okay, so this is now a papilla, and this region what you see here, so this exactly is the medulla region. Okay, so this is the medulla region of the kidney. So full syllabus only for term 2, right? Yes, I'll do it on term 2 full syllabus itself. Okay. Okay, so now once this is done, so you can see pyramids are drawn, then medulla is drawn, cortex is drawn, and the papilla is the apex of this one. So this blue color one what I have drawn, so that is exactly the papilla. So now how you can show the ureter is, the ureter is basically, so now let's see this structure. So from all of this, a papilla, or sorry, not the papilla, pelvis is arising. So slightly extended portion. So now the pelvis. So this part what you see. So this is a renal pelvis which is forming the ureter. Okay. So now this part is actually the ureter part. Okay. So this is the ureter part, and this ending what you see. So this is the pelvis part. So this is a renal pelvis. So once the urine formation occurs, so all that urine will be first entering the pelvic area. So that is the pelvis area. So you can see they are all completely in association. So here I have shown you some gaps actually. So you can actually draw it completely in association as such also. Okay, so better to show this association. So this is completely covering this. So this one is the renal pelvis. Formation is basically by expanded end of the ureter. So now ureter itself is expanded as such, and that is forming the renal pelvis. Okay, so this is the structure. So arrangement in renal cortex is basically the Bowman's capsule, PCT, DCT. So this can be Malpighian capsule also. Okay, so then DCT is there. So that is why a dotted appearance you can see. In the medulla region, this renal pyramid what is there. So this is made up of Henle's loop will be there. Okay, so there is a Henle's loop, and also there is a collecting duct. So because of that, the line like appearance we can see in this region. Okay, so this is with respect to the renal pelvis, ureter, Henle's loop, or a collecting duct. Okay, so now nephron structure. Let's have a look at the structure of a nephron. So I'll just draw it faster. Just a second. Okay. Now this is actually the Bowman's capsule. Okay, so this is a Bowman's capsule. So you will be showing a PCT over here. So this one is a PCT. Okay, so then you will continue down. So there is a Henle's loop. So this one over here is a Henle. Okay, 1 second. So yeah, so anyway, I'll just continue as such itself. So now this one is all right, just a second. It became too thin. Okay, so now Henle's loop when we draw. Okay, so this is the Henle's loop which we need to show. So then there is a PCT, DCT formation again. So there is this structure. So there is a formation of a PCT and DCT, and then you will be showing a collecting duct. Okay, so here you can see a collecting duct. So this collecting duct is opening at many other ends also, because it is taking a urine from other nephrons as well. Okay, so now this is a collecting duct. So we will just label this one. So this is a DCT, this one is a PCT, and this part what you see, so this is a Henle's loop. Okay, so then what you need to show, so when you are drawing afferent and efferent arteriole, make sure that, just a second, make sure that you are drawing a wider diameter for the afferent arteriole. So now this is the afferent arteriole, so then it will be joining here. So you know efferent arteriole is leaving, so here is the efferent arteriole, so here the narrow lumen will be there. So you can see the difference with respect to this one, the diameter or wider lumen you can find at the afferent arteriole, and here the lumen is less. Okay, so narrow lumen, so this is the efferent arteriole, because the space is less over here. Because of that, since the volume is less, higher pressure will be generated. So you know that the volume and the pressure, so they are inversely proportional. So now volume is less, the pressure is going to increase. So because of that only ultrafiltration process will occur. So ultrafiltration will pressure will, the filtration process will occur because of the higher hydrostatic pressure that is generated. So whenever you are drawing, make sure that you are showing this difference. Okay, so narrow lumen has to be shown at the efferent arteriole. So then you will be making a mass of a capillary over here. So this is basically the capillary formation. So glomerulus, you can just draw it randomly. So this is now a glomerulus. Okay, so this is a glomerulus, this one is a Bowman's capsule. Okay, so this is a Bowman's capsule that you will be showing here, and both of these two together, so we call it as a Malpighian capsule. So this one is a Malpighian capsule. So I'll just erase this part. So this is Malpighian capsule. Okay, then what you need to do is, so here once it is drawn, so make sure that you'll bring it down here. Okay, so that is basically again a secondary set of capillary formation has to occur. So you will show the capillaries as such here. Okay, so here the formation of secondary set of capillary is occurring. So this one is a capillary. Now again all of them are joining back, so then you will be drawing it as such here. Okay, so here you will show this structure. So again there is a formation of a capillary at this side. Okay, so you can show the capillary over here. So now one second. Okay, so this will continue down here. Okay, so this will continue, and again here we need to show the capillaries. Okay, so secondary set of capillary where the absorption will occur. So this is the structure of a nephron, so you need to show this capillary, and then all of these will join and they'll be forming, they'll be joining the renal vein. So this is to the renal vein. Okay, so this is coming from the renal artery. So even if we have to show, so this is coming from the renal artery itself. So this will be the renal artery which is dividing and forming this structure. So that is exactly the afferent arteriole. Okay, so this you already know. So that is the structure that I have shown. So you can make a note here, the diameter is wider and here the narrow lumen is present. So here wider lumen, narrow lumen is there. So because of that higher hydrostatic pressure is generated. So that you need to show. Okay, any doubt in this? So neuron structure. Okay, so neuron structure, what is the time? Actually I have another class to go, so maybe a remaining part I can complete in another session. Okay, so okay, I'll complete this one. Okay, yeah, so now you can see the neuron here. So neuron, when we are drawing, again make sure that you are showing a neurolemma. So let's see, this is the neuron part now. Okay, so here is this neuron. One second, it went wrong. Okay, so now you can draw the neuron as such. So there is a neuron, all right. So one long process you need to show. So this would be the long process of the neuron, and all these are short processes which will be ending up with the dendrites. So you can just show the dendrites as such. So now these are all the dendrites for associated with the dendron. So there is, this is now a dendrite. Okay, so we will in the center we will draw the nucleus and smaller nissl granules that you need to show throughout the cytoplasm. So smaller nissl granules are present. So nissl granules are actually the breakdown products of a endoplasmic reticulum. Okay, so this is a nissl granule, and here is this nucleus. Then this one is a dendrite. So dendrite is there. So these curvatures, what you find, so these curvatures actually the dendrons. So I'll just show it, label it here. Okay, so this part is the dendron, and this whole part, so this whole part, what is there? So this is exactly the cyton, we can call it, or we can also call it as a cell body, or this can also be called as a pericardium. So this can also be mentioned as a pericardium. Okay, and after that we need to show the myelin sheath and other things, that is neurolemma. So here is the myelin sheath. So I'll just take the different color. Okay, so here is the myelin sheath which is lining on both the sides of this axon. Okay, so both the sides of our axon is lined by this myelin sheath. So then there is a neurolemma which is covering this myelin sheath. So we will show this neurolemma here. So there is a covering for this one. Wherever myelin sheath is not there, but the neurolemma is anyway covering this structure. Okay, so here you do not find, so this junction what I am showing them, so there the myelin sheath is absent, and those places can be called as a node of Ranvier. So wherever this myelin sheath, so you can see that the myelin sheath is not there, but only the neurolemma is there. So these spots which are there, so these are called as a node of Ranvier. Okay, so node of Ranvier is there. So this red one which is there, so that is exactly the myelin sheath. So we will just label this one here. So this is the myelin sheath, and covering the myelin sheath you see one more membrane, so that is the neurolemma. Okay, so this is the neurolemma, and axon ending we need to show here. So axon ending is basically this part. So this one is the axon ending which can have a bulb at the end. So this is the axon bulb, or we can also call it as a axon terminal. Okay, so axon terminal is there. Okay, so axon terminals are having a axon bulb. So basically neurotransmitter will be stored in there. Okay, so this structure is very easy. So there is nothing more. So nissl granule, pericardium, axon, I'll just label that one. Dendron, dendrite you saw. Then there is a neurolemma. So in between neurolemma, no, you can just show here a nucleus, you can show in between over here. Okay, so associated with this you can just show a nucleus also as such. Even if you don't show, that is fine. Okay, so one nucleus at least if you show, so that's well and good. So here you can just show a nucleus. Okay, so now nucleus you have drawn, node of Ranvier is drawn, so every other thing is drawn. So axon I haven't labeled, so this long process which is in, so that itself you can consider it to be a axon. So axon is again covered by this sheet. Okay, yes, any doubt in this? So this is the brain structure. Okay, so brain structure, what you can do is make sure that you are drawing the convolutions there. Okay, so let's just draw the brain as such. So this is the brain now. Okay, so then you can show this folding as such over here. Okay, so then you can make this particular structure, a pons can be made at this point, then you can continue down here. Okay, So now this is the structure. So brain stem is there. So here you can see, as you can see in this, so there is a furrow formation. So you can make a furrows over here. So somewhat disturbed furrows that you need to draw. Okay. So not a plain furrows. So all you need to do is disturbed furrows. Okay. So this is now a furrows which you can show it as such. Okay. So there are furrows as such. So this need to be as disturbed, like not a straight line. So this is the one that you will be making. So like this you will make. Okay. So after that, so this is the cerebellum is ready here. Okay. So one more we can draw in here. So this is a disturbed furrow that you will be showing. So this is the cerebellum. So this part is the pons, and this part you need to show the convolution. So we will just make the convolutions as such. So these are the convolutions that you can show in here. All right. So these are the convolutions which are present. One second. Okay. So one more I can draw. So folding. So convolution is nothing but a folding will be there. Okay. So folding you can. So if they are asking you to show the meninges also, so in that case you can show the meninges also. So just you need to draw the pyometer, arachnoid, and then the dura mater. So if they are asking you to show this, so pyometer you can just show the foldings along with this one. So this one can be the pyometer there. Okay. So pyometer also is somewhat folded along with the brain. Okay. So this will continue down there. So this part, what is this? So this is exactly the pyometer part. Okay. So other than pyometer, so you can also draw the arachnoid. So now in the center, so you need to show the corpus callosum. So just make a layer as such. So here you can draw hypothalamus like. Okay. So then one more part that you can show. So hanging from the hypothalamus part. Okay. So here you can show this pituitary gland as such. So this is the pituitary gland as such you can show. And along with that there is a corpus callosum that you need to draw. So what you can do, so you can just connect it as such. So I'll just change this color. So you can draw one corpus callosum here for drawing this one. Okay, 1 second. So this got spoiled. Okay, so one corpus callosum that you will be showing, the one which is connecting both the sides. So just draw the lines as such over here. So this is the brain structure. So you have almost labeled every part, drawn every part. So corpus callosum, hypothalamus, pituitary gland is shown, cerebellum part, and spinal cord part is also shown. So here you can draw more of foldings can be drawn. So you can just draw two more foldings as such. So just go on drawing the foldings over here inside. Okay. So that the complete brain you can show in here. Okay. So this just draw some sulci and gyres you can show in here. So this will be the structure of a brain that you can draw. So thalamus, hypothalamus. So this is the thalamus part, and again you can come up with the hypothalamus part here. So then the one which is hanging from here. So this is a pituitary gland. So then what you need to show is the cerebellum part. So this is the cerebellum part. Then this part what you see, so this is a cerebrum. Okay. So cerebrum that you will be showing. Then this part is the corpus callosum. Okay, so this is a corpus callosum. So this is a brain stem or medulla oblongata also can be mentioned. Sorry, this one I'll just show. So here I'll show this brain stem or medulla oblongata, and this part I'll just label it as the spinal cord. So this will continue down there. So this can be the spinal cord. Okay, so you can just label this as a spinal cord, and this part is a pons. Okay. So since we are done, so this one anyway I haven't labeled. So this is a pyometer that I have drawn. So you can you can draw two more coverings as such. So if they are asking you to draw, so you can draw the arachnoid and dura mater also. So this can be one more covering you need to draw. So there is a dura mater. All right, then there is arachnoid you will be showing. Okay, so the outermost innermost here is the pyometer, and if you want to show the skull also, that is cranium box, so you can just draw one more covering above this one. Okay, so there is one more covering, so that can be the cranium part. Okay, so again if you want to show, you know, the cerebrospinal fluid. So in between these you need to do the filling. So this can the fluid which is there. So over here, so that can be the cerebrospinal fluid. So we will just label this one, cerebrospinal fluid can be there. Okay, so you can just show this one. Ma'am, do you remember skies? Why not? I remember skies. I worked there. Okay. So this is Yash. Yash. Okay, so I remember you also. You were in class 6 when I was teaching there, right? Class 6 or no, class 7 you were in. Yes. Okay. So since we are done with today's timing, okay. So before I continue with the last part, so these parts are there. So I'll just show you in the next classes also. Eye structure, ear structure, you know, and some more structures that I need to show. So that is with respect to spinal cord. So I'll come up with one more session and I'll draw these remaining diagrams. Okay. So since I have a class now, I need to end this one. Okay. So see you all in the next class. So before that, there is a champions batch, all subject batch. So this is for ICSE class 10. So if you are subscribing for ICSE class 10, so you will be getting subscription absolutely free for class 11 and 12 also. A new batch is coming, a warriors batch. It is a all subject batch. Again, you know, the mode of delivery is English over here. So March 16 it is starting today itself. Okay, so you can have access to these batches. What you need to do is go for the subscription of Unacademy and you can use my code Vani YT. So with this referral code you will be getting 20% discount on the subscription. Okay, so you can use this code and there is an offer that is going on. So that is why I'm saying only 20%, or otherwise it is usually only 10% will be there, but because of the offer you have a 20% off today and tomorrow. So go for the subscription today and you know you can use this referral code Vani YT to get a discount. So Bucks Bounty, opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Okay, so how you can watch my special classes? So download the Unacademy Learner app, open the app and click on get started. Then you can enter your mobile number and the OTP, then sign up using your email ID. So select class 6 to 12, select the category ICSE that you are in. Okay, so then select the preferred language. So once you are done with that. Okay, so you can find the description link in the description. So you can click on the special class link which is available, or what you can do is directly you can go to the Play Store, download the Unacademy Learner app and follow this. So you will easily find the different educators and their classes. Okay, so other than that you can use my code Vani YT. Okay, so there is a code Vani YT, so you can use this code to get 20% discount on the subscription, as there is a offer today and tomorrow. Okay, so thank you all. Before I end the session, so you can you can press on the bell icon to get notified. Subscribe to our channel, that is Unacademy ICSE and ISC both. Like and share the video and download the Unacademy Learners app. Okay, so see you all in the next session. Bye bye everyone. Thank you so much.